Yeah, so if everything was self-explanatory about the GLM, then you really have the backbone of all of the, like most of the fMRI analyses that people do already set. Um, the idea then is that, we've now analyzed the single voxel. We saw how we could create like an expected time course from what we think is neural activity and then convolve that with a bold response and then see whether that expectation fits with our model using this GLM fitting procedure. And based on that, we can also get statistics, right? We can get p-values. And of course, as scientists, all we ever want is p-values. So that's, you're done. You're done. Um, no, but seriously, that is the most important part is of running an fMRI analysis on your own data set is to repeat that same analysis that you now know to do on a single voxel across a whole brain, right? So they're all, for the intents and purposes of this type of analysis, they're all the same. So it's just a parallel problem and we just run it. So that's good that we've learned that, but it sort of assumes that you're looking at a voxel and that voxel is measuring one specific location inside someone's brain, right? But that's not the type of data that comes out of the scanner. People move, for example, and as you move, the scanner doesn't move the voxels with your head. You're actually moving your head through the box of voxels that the scanner is recording. So if we just say, oh, we're just going to like record this, look at this single voxel, it's not one single location in the brain, right? We need to make sure that this voxel is recording the same location throughout the experiment. And there's a whole bunch of things that relate to, do we know where we're recording things from that we need to establish? This entails performing a lot of different steps of analysis, and that's what I'm going to talk about today. This is this whole pipeline of pre-processing steps that we need to do to be able to analyze our data in the way that you already know how to. And this is incredibly complex. It's also talked about in the lectures on YouTube a bit, but I'm going to give it my own sort of interpretation, what I think is important in these. So I'm going to go through the basic steps, but rather quickly. And then what I want to highlight is what is the modern way of going about this. And there is a very distinct, modern, reproducible science way of going about this that I'm going to explain, and that is also the topic of this week's practical. So the practical is online, and it's going to show you how this pre-processing works in terms of the results that it gives you. And what we're teaching you with this practical for this week is how to interpret these. So when is a subject good? When is a subject bad? What does that type of thing correspond to? If there's something wrong with my data, is that something that is due to the subject misbehaving, or is it due to the scanner misbehaving? All of that sort of thing you'll be able to sort of interpret and analyze by yourself after this week's practical. But let me first just start showing, like sharing my screen and all that sort of stuff. You can see this, right? Yeah, I see some nods. Great, thanks, Ines.
Welcome.
Yes, so there is a long road going from raw data to the results that you want, and this road is shaped as a sort of pipeline. So we have this pipeline of analysis steps that we need to go through in order to be able to interpret what our results are. And for example, there's these things like I just referred to. We need to know that we're looking at the same location uh, in the brain all the time, right? How do we even, like, if people move, how do we fix that? Let me see. I have to admit a few people. There you go. Great. So we record this beautiful 3D box of data, and we record it, let's say, every second. Um, the scanner reconstructs that from the radio signals into an image for us. But then the data that come out are horrible. They are horrible. They are distorted. There's movement. There's differences in when the different parts of the images were acquired. There's really weird slow drifts due to the fact that the scanner is heating up and your subject is becoming tired. And there's a like it's crap. And what we want is to fix that. And then one important thing that we also need to realize is when we fix that, how much have we actually fixed and how much of the problem still remains? Right. So one of the important things is when we go through an entire pre-processing pipeline that sort of massages our data into the right format so that it sort of corresponds to what we expect it to correspond to. That sort of thing is never perfect. And we want to always inspect the results at each of these different steps to know how it isn't perfect. So I first want to go into a few basics. The YouTube lectures go into a bit more detail here, but for the intents and purposes of these slides, I wanted to just get a few ideas out of the way so that we can build upon those. So the first is the idea of linear transformation. So this is my former PhD student, Dan, who made these slides. And I'm going to use this image of Dan as an analogy to an fMRI image and show you linear transformations of his picture. Yeah. So there's a bunch of different transformations that we could perform on his picture. We could rotate him, right? We could scale him, we could shear him, and we could translate him. And all of these transformations are what we call affine, or for our purposes now, linear, which means that they assume a rigid body so that there aren't any deformations here. So this type of transformation of an image, and to be clear, I'm using a 2D image as an example here, but the fMRI images are 3D, but the same logic holds. Yeah, so also a 3D image can be transformed linearly as a rigid body, and that can then be expressed in transformation matrices. So for 2D, this affine transformation type thing, you need a 3x3 matrix. And if you have a 3D image, you need a 4x4 matrix. So if you rotate an image, you have these cosines and sines of some angle. And if you take the 2D points and you multiply them with this matrix and take only the first two resulting dimensions, then you get the rotation. So similarly, if you want to scale something, you just use those two values and the rest you leave to 0, and you just, the same thing goes, you just multiply with this matrix. Similarly for shear, you have these that you multiply and these remain 1, and translation is just here. And you can imagine that if you put them together, you could do something like a combination of a scale and a rotation. And that's true. You can just add these matrices together and then perform some combination of these operations. Yeah. And so here you see again that representing all of these images just as numbers, as arrays, we can start to use very simple sort of linear algebra matrix multiplication things to work with our data. Yeah. Okay. those are linear transformations, and we'll see some uses of those in a bit, right? But I just wanted to highlight those concepts. You assume that in essence, that the shape of the thing, that the image that you're looking at doesn't change. But there are also nonlinear transformations, and those are, I'm going to highlight exactly with the same figure, but nonlinear transformation does change the internal metrics. So you cannot express this as a simple matrix. You have to actually designate for each point in the image where exactly will it end up in the transformed image. So a nonlinear transformation is not as easy to describe. And here are some examples of what you could do. Right, you could pinch or twirl or wave or zigzag. These are just Photoshop filters, of course. And you would express these not as a matrix, but as a warp field. And so, for example, the pinch would be a warp field like this. It tells you for every point in the original where where it should end up in the deformed image. So it's similar to a linear transformation, but it's more complex, and therefore you need this entire field to describe its properties. You could also do the same for, like, you have a twirl as an example, right? There's some wavy stuff. And I think the zigzag here is wrong. It's also wavy stuff. But you can imagine that there would be warp field. That would be a zigzag warp field, right? That tells you what the zigzagged version of the original image would be. So these are two types of transformations. One is simple. Right? It assumes that the thing that you're describing with the image doesn't change shape in any drastic form, whereas these nonlinear transformations, they can very drastically change the image. Yeah, and in principle, they could warp it to oblivion. Right? There is no limit to what you could do with a warp field. It could be completely weird. Right? You could really randomize the entire object. So it's important to realize that when you apply a nonlinear transformation, weird stuff can happen in principle. Yeah? So it's use these with caution sort of thing. And I'm immediately going to go into one use of this nonlinear type of transformation, because when data come out of the scanner, they are distorted. And the reason that they are distorted is, well, we try to encode space using frequencies, right, with a gradient that we set. One of the YouTube lectures also talks about this. We do the same for phase, but because the B0 field is not homogeneous, the encoding of space isn't as good as we would like. So depending on how the B0 field is distorted, and it's already distorted if you put someone in the scanner, that's going to warp our representation of space. So we think that space is linear, but because of these distortions in the magnetic field, it's not. And this is something that we have to work around, that we have to realize and that we have to work around. Because when you think that something is linear, but it is not under the hood, you get warpings of images. And what we end up doing is we want to know how this B0 field is distorted. So what we, for example, can do is record a B0 map. So map out the distortions in the magnetic field. It gives you something like this. So it tells you where the B0 field is distorted. And then we can correct this original image, correct how it must be distorted, and then get a corrected or unwarped image. Yeah. There's a bunch of different ways in which you can do this. You can get the ground truth B0 map. You could also create two different images that have opposite warpings and then sort of average them together. So find the middle ground between two oppositely warped images, and that can give you the unwarped image. But as I've just explained, right, this is a nonlinear transformation. You need to actually really create a warp field to make the EPI image, your functional images, fit onto, for example, the veridical anatomy. If you wouldn't unwarp, it means that these functional images don't live in the same space as the anatomy that you get. Usually anatomical images aren't distorted at least this much. Right? So they're minimally distorted, and this makes them fit, right? It makes the functional images and the anatomical images fit together, and that's what we want. Otherwise, you get activations outside the brain, for example, somewhere in front of someone's forehead, and that sort of thing is just impossible, right? We need to know where things are, and this is a necessary step. And it can be pretty drastic, these sorts of things. This is even still moderate. This is from a 3T scanner, at a 7T scanner. These things can be much, much worse. You really see that here visual cortex shifts at least a couple of voxels. Yeah, so if you thought something was in visual cortex, it wasn't. Whoop. And for example, you see the frontal cortex also moving up and down, like in and out, very strongly. Yeah. And this is worse exactly in regions where you have very large field distortions, and they happen mostly where the air hits the tissue, so somewhere near the sinuses. You see that there's a lot of shifts here, and not that many in different places and other places in the brain. And so you get strong shifts where you're close to air. So we can fix those now using this unwarping. And another thing that we need to make sure of is that when we record all of these images over time, that they're actually in the same location. So people always move. It's really hard to keep people still. Even really, really well-trained subjects still move because they breathe, for example, right? And their hearts still beat, and that means that their brains move inside their skulls. So there's really no getting around this. And if you look at this over time, it can really mean that people sort of sag away, and there's like a really, really big discrepancy between a voxel that you started out recording, which is, let's for example say we're starting out to record this voxel. Well, at the end of the acquisition, that's in white matter, right? So you're really moving from a region of the brain where you expect some signal into some region where you don't expect any signal. So we need to correct for this motion, right? Because only then can we pick a voxel and say, this relates to one single location in the brain. Otherwise, it's just shifted around all the time. We can't have that. So what we can do is we can calculate how much we would need to shift and rotate and translate one image relative to the other. By just fitting, right? This is a fitting problem. You can fit these parameters. And so you can find how much you need to shift and rotate an image to fit another image. And you could do that for all of the time points separately. So you do that for each of the time points. Here, we're using as a target the middle TR, right? So the middle time point. You can choose any reference. You could choose the first. You could average the whole thing over time, and so forth and so forth. Yeah. But the point is, if you have all of these translation matrices, these linear transformations, then you can apply them to your images, and then they all reside in the same space. Otherwise, they do not. Yeah? So we're already talking about two ways in which the data that we thought we were recording, like nice voxels, very precise, this and that, it doesn't make any sense, right? It's not even inside the brain sometimes, and then it moves all the time. So we need to correct for these different things, right? And this is what we do. We can just put all of these brains back into the same space. So one way of illustrating this would be to just show you a movie, and this is what I'm doing, right? This is just a section through the brain in the axial direction, right? That's this slice. So on the left, you see before movement, and this is exaggerated movement. This is an experiment where we ask someone to move quite a lot. And on the right, you see the same scan, but after the motion correction. And you definitely see a large difference between the two versions, right? Now, later I'll tell you that this is not the end of it, but that is after I recap a bit of the GLM that we went through last week in the practical. So let's say you have a bold signal. So what I'm going to do is quickly show you how the GLM works again, and then go back to how we can fix motion. Yeah, then we'll go back to that. So let's say you have a bullet response, right? It is like this. It's noisy, and then sometimes it goes up. Um, and there's apparently 3 trials with a large response and 3 trials with a not so large response. You see that? This is something that's very similar to what you already analyzed in the practical this week. Yeah. So this must look rather familiar. So if we fit our GLM, what we get is, of course, a bunch of beta weights that you all know and love. We get a beta weight for the intercept, right? We had to add it because otherwise we wouldn't be fitting this offset. And we have a beta weight for these trials, and we have a beta weight for these trials. Clear, right? We know this. This is what you did this week. So the GLM allows us to fit these beta weights and find how strong the response was to this part of the experiment and to this part of the experiment. And that is quantified by these beta weights. All right, so here the values for these beta weights are 50, 10, and 5. That's what you'd get out. What I want to talk about is something that that you also saw in the practical, right? What you're doing to get a t-value is take the standard error and then use that to divide the beta weights, right? So in principle, and this is not entirely, I'm simplifying things here because I want to make a very simple point, and then we can go into how we go further. You look at how much your model explains in terms of variance, how much your data explains. This is the part of the, you take the squared sum of the residuals, right? The part of the data that you do not explain. And this gives you this standard error of the beta weights. Now, what you have to realize here is that the better you explain the data, right, the fewer or the lower the values are that put space between the data and the model. Right? If you have a perfect model, this will go to 0. You square 0, it's still 0. You sum zeros, it's still 0. Right? So a perfect model gives you a standard error of 0, and then your t-statistic, as you see this going to 0, this goes to infinity. Yeah. So what you want is a model that explains your data really, really well, so that this goes to 0, that the squared sum of all these goes to 0, and that your t-stats go up, because then your p-value goes down. Right? That's the essence of what the final parts of your of the practical last week. So that's the logic, right? We want to explain the data as best we can. And these are the parts that go into this, right? So this is the data here figured in blue. The best fitting model is red. And then we have this bunch of residuals. Now this set of residuals is our arch enemy, right? We want these all to die. We want them all to go to 0. So in this case, it's pretty okay. The residuals aren't that big, but in this case here, you're looking at much noisier data, right? So that means that the squared sum of the sum of squares of this is much higher than it is on the left, right? And what was a nicely statistically significant beta value here, that identical beta value here gives you a problem, no longer a significant result. Yeah? So really, whether you find something in fMRI depends on these residuals, right? Our arch enemy. If you manage to explain the signal better, it will decrease your residuals and thereby give you the opportunity to find something where you could not before. What that means is that if you find some structure in the residuals that you manage to explain, you're golden, right? That's what you want to be doing. So having said that, we go back to the motion parameters, right? So we were putting the branes on top of one another, time point by time point, and we actually have a measure of how much we needed to shift the brane from time point to align. Right? So if some of these residuals, so some of the unexplained signal is due to head motion, and in many cases it is, then we can actually create a regressor based on these motion signals that will allow us to explain parts of the variation in the signal that we couldn't explain before. And that will allow us to decrease the residual variation and thereby improve our model. Yeah, we call this a nuisance regression analysis. Yeah, so you just add these regressors to your original design matrix, and that will allow you to capture variations in the signal that maybe you're not interested in, but they would have become part of unexplained signal, and you hate that, remember? So that's the point.
Thomas, I think there's a couple of questions in the chat.
Oh, I'm missing those. Ah, I'm so sorry.
I can
Yeah,
ask my question not in the chat maybe, because I didn't understand in the formula before how data and model are different, because I would think that the model is derived from the data. So how can you then subtract them from each other if...
Yeah, that's a good point. So let me make that clear. Oh, I'm in the wrong slide. Sorry, the chat the window, I missed it completely. In any case, yeah. So how are data and model different, right? Well, the GLM that we did, we had these different regressors, right? So different time courses of a possible signal, and we fit those to the data. Yeah, so we get this y hat, we called it in the practical. You remember? Cool.
Yes.
Great. So here, that's what we call model here. Right? So that's y hat. It is the part of the signal that we explain. So that's this. And that's y hat. So the difference between the part of the signal that we can explain and the data is the part that we cannot explain, right? Because the data is just the data. It's fixed. We can't really change anything about it.
Okay, so model is basically the predicted value.
Yes.
And data is the observed value.
Yes, you have observed value, that's data, and model is the predicted value based on the best fitting model.
Oh, okay, perfect. Thank you.
Yeah. So how do we come up with a model?
You just answered it basically. So I had the same question. Yeah.
Great. Yes. Yeah, very important. So when you see model here, think y hat. But just to be clear, right? The difference between low residuals and high residuals, and that we hate residuals, that's clear, right? And you get the logic of having large residuals deflating your t statistic. All of that's clear. Good.
Sorry, I would have a question to the slide before.
Yeah.
The first one.
This one?
That one, yeah. I didn't get exactly why the beta values are. What was the first one? 50, I think?
Yeah.
Yeah.
So 50 is the baseline of the signal, right? So that's the offset that you need to put the signal at to start explaining the signal here. Yeah. So this regressor and this regressor, they're not trying to explain any signal here. So that has to be captured by the offset that we see here.
Mm-hmm.
Yeah. So that's the intercept regressor. And if you look at an fMRI image where you have this type of time course for the entire time course, or so for all voxels, I mean, then if you fit this intercept, that would be the anatomical image in the background, right? It doesn't change over time. That's the essence of the anatomical image. Here, this is 10, of course, because you need to multiply this model time course by the value of 10 to gain this signal that goes into 60, goes on to 60, right? And similarly, this one, these are half of the amplitude of these, so these need to be multiplied by 5 to best explain this signal, yeah? And the NumPy least squares function that we use in the practical allows you to do exactly that, right? It finds these beta weights based on the, yeah, we can go into linear algebra for that, but it's not important. Yeah.
No, I got it. Thank you very much.
Cool. But this, in many different sort of paradigms, this is really important, right? We want to explain the signals that we have as best we can, and how do we go about that, right? And one of the options is to add variables that vary over time to the design matrix in our GLM analysis. So we find beta weights for each of these, but we're not interested in them as such, right? We don't want to know whether a voxel responds to motion to the right. Who cares, right? Motion of the head to the right, that is, not on the screen or whatever. that's not really important. But if the voxel has signals that are explained by this, you don't want to see that variance in the residuals. You want to capture it and throw it away. Clear? Great. So, for example, a spike like this would give you huge transients in the signal, right? So even though we've put everything in the right place with the linear transformations, with the motion correction, we still have all sorts of signals originating from this type of head motion, and we want to take it out. So using these as nuisance regressors allows you to regress this type of effect out of your data and thereby improve your statistics, or in general, just improve the way that you're explaining your data. Yeah. Great stuff. Now, a bunch of these. So this part of the course is always me telling you how bad Fmri is, and I feel a bit guilty because, you know, I'm just going to tell you, you thought that you were measuring something in the brain. Well, it's not neurons, it's blood. You thought you knew where it was because it has such high spatial resolution. You don't. Yeah, so you need to go through all these steps, right? That's really what the pre-processing is for. And I'm just going to go into another one of these things where you thought you were measuring something, but it's much more crappy than you thought it was. And that is, we're measuring blood, remember? So we know that, you know, your heart beats, right? And that's going to have an influence on your blood, right? Also, there was this thing with a bold response having to do with oxygen, right? You breathe, so that's going to change the oxygen in your brain's blood, right? So that's going to have an effect. And then the respiration is even worse because when you breathe, your whole body more or less changes position, right? So breathing actually has a pretty large effect on where your body is in the scanner. And that changes that B0 magnetic field, and it really changes also the intensity of the images that come out. So breathing actually is bad. Yeah, it's really hard to like record Fmri from a dead subject, I realize. But these things are necessary evils. So, okay, that sounds wrong too. But think of them like that. Yeah, your subject's heartbeat and your subject's respiration are necessary evil. What do we do with necessary evils? Well, they're there anyway, right? But as I just showed you, you can use, if you can actually record them, you can start to try to see which part of the data you can explain from these factors, right? And thereby clean up your data. So we can try to measure heartbeat and respiration separately, which we do with this PPU, pulse perimeter oximetry type thing, right? So you can, it's like an infrared LED and it measures the heartbeat. And you have these sort of balloons that you see here where we can measure someone's respiration. And that allows us, so you get these signals like a nice cardiac cycle and a nice respiration cycle. And those are then sampled into the slow Fmri signal, right? That's what we're working with. So the Fmri is measured like once every second or so. And you have like the fast cardiac cycle that just noises this up because it's aliased into this slow recording. Yeah, so you're taking these snapshots of your brain's blood, and sometimes it's at the peak of the heartbeat, and sometimes it's at the trough of the heartbeat, and that's going to cause a difference in your data, and that's what we call aliasing in this sense. So there are methods with which you can then create these nuisance regressors from these signals. It's called retro ICOR. It's not really important how this works for these purposes. But it stands for retrospective image correction. So what you can then do is similar to that motion correction thing where you have a signal, you can use these as regressors. So you can regress the effect of heartbeat and respiration out. And so this allows you to clean up your data quite tremendously. Now, one other factor that we have, and after this we should have a break at some point. One other factor is that sometimes we have slow drifts in our data. So you can have these very, very slow drifts. For example, if people are starting to fall asleep in your experiment, you can imagine that the signals change. But the scanner can also heat up because it's scanning so hard. And that can also cause all sorts of changes to your signal. So one important thing to realize here, and this also harks back to the case-based thing of the images that we record, is that all signals that we measure, so also the time signal of an fMRI voxel, you can express as a combination of sine waves, right? So in this case, you have this pretty complex looking signal, which is actually the superimposition of 4 different sine waves of different frequencies. Yeah? And we know from Joseph Fourier's mathematics that we can express the signal as this combination of sine waves and cosine waves, where one way of looking at this signal is through as a power spectrum. So that's the amount of power that resides at each of these frequencies. So this is one way of describing this, this signal. So this has a pretty strong power at 5 hertz in this case, apparently. So how does this apply to fMRI data as we look at it? Well, what you can have is a voxel that has a very slow drift in it, and it has an offset other than 0. And what you then find is that these very, very low frequencies have very, very high power. We don't want that. So just to clarify, we're not interested in these slow drifts. This is something that we could do without. We want to do without. This is like, we're just in this fMRI experiment where, for example, we're doing a working memory experiment and things change on every trial. We're interested in that. But this is much slower than the parts that we're interested in. So what we can do, so this part of the data we don't really care about. So, and that is these low frequencies. So what we want to do is take out these low frequencies and get a high pass filtered time course so that we can start looking at the power at all the frequencies that we do care about, right? These faster things at which the experiment happens. So that's what high pass filtering does. It lets the high frequencies pass and it deletes the lower frequencies. Yeah. So this is just in general what you always want to do. Sometimes people filter their data, but there's a different sort of paradigm to do this in. What you could do is take very, very slow sine waves. In this case, we would call that a cosine basis set. That's not too important for now. But those we can just put into our GLM as regressors that would capture slow drifts. Right? So the same trick, we can regress all of this out. Yeah, so in this case, this slow drift would be something of a residual, right? If we use our standard design matrix, we'd get this slow drift and that would be a residual. This slow cosine basis set would allow us to capture that, in which case it's no longer unexplained signal, right? And that's what you want. You want, like motion, like heartbeat, these slow drifts, you will just want to be able to regress them out and not have them count as unexplained variance. Like I said, unexplained variance is your mortal enemy. You want to kill it, right? So if you can capture it using a slow drift basis set or a motion correction or physiology, that's what you do. Now I do want to add here that there's sometimes a problem, right? When I simplified the GLM analysis equation just now, when I did that GLM recap, I didn't say anything about degrees of freedom, right? So in that standard error, I said, like, I'm making this too simple. Yeah, that's due to the standard error that I left out. So the logic here is that you could say, but then I can add physiology regressors. I can add motion regressors. I can add slow drift regressors. I can add like 500,000 regressors, right? But even though they might all capture variance, it starts defeating the purpose, because if you're interested in something like a t-statistic, Right? That is built up from this standard error. And one part that I left out before, but that is essential here, is that this degrees of freedom is something that changes this standard error that we were talking about. And so the larger the degrees of freedom, that's the residual degrees of freedom, the smaller the standard error on your estimates is. Now, this degrees of freedom, it's composed of the amount of time points in your data minus the amount of regressors in your design matrix. So the more regressors you put into your design matrix, the lower the degrees of freedom, until at some point it becomes so small that this factor starts to explode, right? And there goes your t-statistic, and there goes your p-value. Yeah. So there is a trade-off. You want to add regressors to your design matrix because it allows you to capture unexplained signal, but for your statistics, it will start biting you in the ass. Yeah. So there is an optimum somewhere. Yeah. And so think of this, if you're an AI student, this is really a fitting problem, right? In which case, if you're adding too many of these nuisance regressors to your design matrix, what you're doing is you're overfitting. Yeah, so the analogy is perfect here. So there is no free lunch. Let's take a break. Why don't we reconvene here at, well, 2:30. This is supposed to last until 3:15, right? So we'll have a nice and long break. We'll pretend that this is an actual lecture, 45 minutes and then 45 minutes with a 15-minute break. What do you think? Great. So go get a coffee. I'll see you right back. Oh, yeah. Let's get going. There are a few questions, or one mainly, about how the Fourier transform relates to the using the slow regressors in a GLM design. So they're more or less the same thing. If you were to perform a GLM with only these very slow sines and cosines, and then take the residuals, what you've in essence done is your high pass filtering. So the two trade off. Yeah, so you can see the Fourier transform, you can implement the Fourier transform as a GLM. they really are more or less the same thing, because we can see each time varying signal as a combination of sines and cosines, right? So it's a way of translating into one another. And the GLM allows you to do that too, but it also allows you to encode the parts of your experiment that you're interested in at the same time. So you can combine this Fourier type quality of the GLM with the experimental analysis part of the GLM. They can all fold into one single fitting routine. Yeah, like Leo says, it's a time series thing. So yeah, that's that. What I'm going to do is share my screen and continue the lecture, because I think there's a few things that I wouldn't want to miss, or I mean, wouldn't want you to miss. So, one final thing is a sort of caveat that I want to talk about, and then we're more or less done with the like how do we pre-process things. So that means that you would be able to start looking at the outputs of the pre-processing streams that we're going to be using in this course. Okay, so spatial interpolation, what the hell is that? Well, let me show you. Let's say you have an image and you have a bunch of pixel values. Let's say these are your pixel values. So for example, in motion correction, where you calculate how much you need to shift an image to align it back to its original location, you might have to shift it some portion. Let's say this, like half-half. Yeah. So then we need to regrid everything, right? So we still want to have the same number of voxels in all directions, right? And if we don't do this, we haven't actually changed the data. So we need to change the data. This means that we need to recalculate the values for each of the voxels, right? So what do we do with inside this red box? Right? So one way that you can do this is by taking the average. In this case, that would mean making it the value 24. Yeah? There's a problem here. And this is definitely the case in averaging interpolation, the way that we're doing it here now by creating the value 24. But even with more sophisticated types of interpolation where you do nifty stuff, so that's the sync method that's in the title, you lose information. Because if you do this over and over again, you end up smoothing your image. You end up taking averages, taking averages of average, and so forth and so forth. Now, why is this a problem? Why am I talking about it here? Well, you start out with this really nice and crisp image, and then you're going to do motion correction, you're going to do unwarping, and then you're going to co-register everything to your average subject, for example, right? So I'll be talking about that subject in a bit, but what that means is you're going to do 3 interpolations, you're going to do 3 transformations. And if you do 3, you're going to end up having smoothed your data incredibly. Yeah? So what you want to do is avoid this interpolation step, but calculate the motion correction parameters, calculate the warp field, and calculate the warp field to go to some average subject, which I'll talk about in a bit, and combine them all in one single transformation step, and then perform one single interpolation. That's really what you want to do. Right? So the pre-processing pipelines that we'll be inspecting, that we'll be talking about, do exactly this. And this is the way to do it. And in many labs, it used to be the case that they would just do this, in different steps. And thereby, what you in essence do is you throw away a lot of information. So that's something that is improving drastically in MRI practice. So one thing that also used to be a thing is people would smooth their data. This is the olden days. We don't. I think we generally would now frown upon this. Because again, even though you might increase SNR, right? So locally you would average away a bunch of noise as you smooth the spatial detail away. What you end up doing is also decreasing the specificity tremendously. So if you look at the earlier Fmri studies, it would be horrible because all of them would just do this, right? So you start with this thing where there's actual anatomical detail and you specific spatial information, and you end up with this blobby shit, right? That's not really something that we do anymore. So you see how many of the different steps that I've talked about until now have to do with the question, what is where, right? We want to make sure that we're looking at a stable spatial location. That's why we do motion correction. That's why we do unwarping. Many of these problems have to do with space. Yeah? And this problem is much more fundamental than people in practice realize, and I want to talk about that a bit. So there's a few points. Oh, yes. Thank you for the full width half maximum thing. Yeah, so even though we might have motion corrected our Fmri results and we may have unwarped them, and then co-registered them to the anatomical of that subject, right? So that's what I'm calling T1 here. That's what we usually use, a T1 weighted anatomical. Then you know exactly where things are, well, exactly to voxel resolution, where are things within the subject, yeah? But the problem becomes when you want to do a correspondence across subjects. Then we start to have an enormous problem. How do we know that the voxel that I'm measuring in my head goes with the voxel that we're measuring in your head, right? And you can think of a bunch of problems that we run into here. So I want to highlight one, is that we're all unique, right? That sounds like a, you know, bathroom tile wisdom sort of thing. But in this sense, it's actually quite problematic, right? Because if you look at different subjects all put in the scanner and measured at exactly the same location in the scanner, they look different, right? This is just a single slice, but it's even worse if you look at the 3 different dimensions, right? So different sizes of heads, different shapes of heads, and so forth and so forth. So it's beautiful that we're all unique, of course, but it's kind of problematic if you want to say something about what function resides where in the brain, right? What do we have in common? So this really sucks. And this was just a gross example, but even in, like, it gets worse the further you zoom in. And specifically, you get that the sulcal folding pattern of the cortex is really, really different from subject to subject if you look at it in the volume. So there's a bunch of different ways that people use to combat this, and I want to highlight a few. So the first is, what we could do is do nonlinear registration to a standard brain, or we could do surface-based registration where we project the functional data to the surface of the brain that I've highlighted before, right? You can look at the surface of the brain. We can project our functional results to the surface of the brain and then say, but you know, on the surface, subjects are more similar than they are in the volume. And I'll explain these different strategies step by step. I'll tell you what the outcome is, though, right now. For the cerebral cortex, you want to do surface-based registration. The cerebral cortex is so variable from subject to subject that the folding patterns don't align well in the volume, even if you've done nonlinear registration, and you're screwed. So you want to do surface-based registration. But for the subcortex, nonlinear registration actually works really well, because there it's an older part of the brain. It's more easily standardized. So those parts of the brain work well for nonlinear registration. So you want to do both volumetric and surface-based registration types. Of course, if you know exactly where you need to look in the brain for each of your subjects, you can do this. You can designate a region of interest and then base your analysis on that. But I won't definitely go into that. So, registration across participants. If we do this in the volume, people tend to use a template, a standard brain, and these standard brain. There are multiple standard brains, which is kind of a misnomer, of course. But the most famous one is the Montreal Neurological Institute standard brain. The standard brain before this standard brain was actually based on a single brain, the Talairac system. If you ever encounter this, the Talairac system, it was based on a single 69-year-old woman's brain, and that was the standard brain. It was just, this is what they called the standard brain, and everyone's brain was warped to this single human brain. Now, this standard brain is built up of an average of, I think in the most recent case, 152 different subjects. So they're all sort of nonlinearly warped together and then averaged in the volume. And you see it looks beautiful, right? It's a nice and symmetric head because you're averaging away all of the subjects' differences. What you also see is that there's some detail here still in the middle, and especially if you take the PDF of this slide and then zoom in it a bit, you really see that there's like, this all looks pretty detailed still, right? You can actually see where things are. But if you look at the cortex here, that's all mushed together. That's like appelmoes. So that's not something that we can use, right? So you really have to think here, where do we still have anatomical detail? Because that's something that we really need. If we want to say, I'm looking at the same location in your brain as I am in my own brain or someone else's brain, then I need to be able to reference to the relevant anatomical detail. So that works in the subcortex in the volumetric sense. It doesn't when you look at the surface. I mean, at the cerebral cortex. So I just want to quickly highlight the sort of processing steps that people do. First, you do a linear registration of a subject's anatomy to this standard brain. So it's more or less in the same location. Right? And then you start calculating a warp field to blow that subject's brain up in specific locations or squish it down or things like that. And so that's usually the way that this type of processing goes. So what you end up with is this a fine transform that you calculate, and then you have this, you calculate a deformation field, and then you apply that. And then you have like the individual subject in the space of the MNI template. That's what you end up with. Right, so this is a multi-stage type of registration. And you can then say, okay, this is in the subjects. So the data come from the subject, but they reside in this sort of average space. So the pros here are, it's really easy to do, and that's why people used it a lot. The con is that, especially for the cortex, like I just said, these gyral and sulcal patterns are really warped into one another. And you get really big deformations, and they don't really make sense. Right? So you can imagine that if you folded the cerebral cortex very strongly, which it is inside our skulls, it's not really, like, it's very hard to get towards this template. Right? You have a lot of individualness in that folding pattern, and it's really hard to capture that in the volume. So this is ugly. So this is more or less what that recipe looks like. If you do this for Fmri, of course, you take your EPI data, so that's your functional data. It's linearly registered to that subject, to the same subject's T1, right? So this is the same brain here. And then this one is nonlinearly warped towards the standard brain template. So this is the template, this is the single subject, this is the functional data. So if you then concatenate these operations, you can actually project the functional data into the standard space. That's the idea. Yeah. Now this is, of course, same pros and cons as for anatomy. This works well for the subcortex, but it doesn't for the cerebral cortex. And So grossly, if you look at, for example, the cerebellum, we still do volumetric stuff there, but that's mainly because the fine spatial detail of the cerebellum is so tiny. We would want to do it at the surface level, but that's just impossible. Our scanners don't have the resolutions yet to actually perform that, that type of analysis the way that we would want it to. So if we're going to do surface-based registration, what we want to do is do some unwarping of the functional data and then register it to the T1, right? So the anatomy of the subject, and that's still within the same subject, right? So you don't have any spatial changes. What we do when we do surface-based registration of the cerebral cortex is we take out this nonlinear warping step. So we take out the standard volume, and we're going to start using the fact that the cerebral cortex is organized as a sheet. Yeah? So those sulcal patterns aren't there for nothing. They are there because the cerebral cortex works as a sheet, right, as a surface, and it's so big that it needs to be crumpled up inside the skull. Right? That's why we have the sulci and gyri. But the functional sort of unit of that brain tissue is the surface. It turns out that you can reconstruct the white and the gray matter. I described this in one of the previous lectures. And then you can blow up someone's hemisphere, like each hemisphere separately, to a ball, right? So you reconstruct the mesh and then blow it up until it's a sphere. You do this separately for each hemisphere. Well, it turns out that if you look at the curvature, so that's the thing that's plotted here on the surface of this ball, of this single hemisphere. So red is inside the sulcus and green is outside, or vice versa, I forget. But it turns out that if everybody is just inflated to a ball, you can just rotate the balls and align them to some standard. And that takes into account someone's folding pattern. And it works really well for the surface. So putting people into alignment on the surface is actually a very powerful approach because it uses the folding information, whereas this nonlinear transformation in the volume does not. So what it actually does is it takes these specific landmarks, like the largest sulci, and places them on top of one another by rotating that ball. And then there are specific variations of this that allow local warpings of that ball. So you can like blow up specific portions of it relative to others. And so one of the ways in which people have started doing this is to take, for example, different types of anatomical information, different types of task MRI, different things like functional connectivity, and then informing this co-registration across subjects using this type of information. Right? So I think I spoke about this before. This is part of the Human Connectome project where they did 1,200 subjects. They scanned 1,200 subjects, and that gave them this sort of atlas of the human brain where they had something like 200 regions in the cerebral cortex that they could really closely align from subject to subject. Yeah? So in this case, you're not just using anatomical detail, you're using functional and anatomical and histological types of information, and you're using it on the surface. So that makes the co-registration between the subjects double better. Is that clear? So if you have any questions about the specifics of this, I'd be very glad to talk about that in detail. For now, I just wanted to show you again that this is more or less where the state of the art is. This allows you to really shoehorn people into the same reference frame, and this really improves the comparisons across subjects also. You can imagine that if you don't really have this correspondence between people aligned this well, you need, for example, twice the number of subjects to get something that's significant from your experiment. Whereas if you can do this, things are much more alike. So that's all good. We've now gone through this enormous sort of pile of different processing steps that we can take. Right? And what I want to highlight now is how it used to be done. And so if you look at the YouTube clips, that's more or less how things used to be done. And as a field, due to the whole reproducibility crisis, we've started to take steps towards more reproducible fMRI. And specifically, people have gone into developing new methods that ensure reproducibility. And today's or this week's practical goes into doing things like quality assurance and this pre-processing pipelining in exactly these reproducible ways. Yeah. Now, you can imagine that given the couple of steps that I highlighted, I think I highlighted something like 10 different pre-processing steps that one would need to do to get to an analyzable data format. Yeah. So these are those motion correction, field map correction, filtering, and so forth and so forth. Now, this is from a review that highlighted the problems with the fact that this is such a complicated sort of machinery that we build before we even start analyzing our data. As you see below here, there's choices that you can make for each of the different steps, right? You can do some different type of motion correction, some different type of field map correction. You know, you have a bunch of different choices that you can take at each of these different steps. And so the total possible workflows that you see here at the bottom is something like 70,000. Yeah? And so it used to be the case that every lab would have its own pre-processing pipeline, which also means that it's very hard to compare different labs' results. It's more or less impossible. So this is really something that caused a lot of problems, right? How can you know if something's reproducible if you know, even the basics aren't doing exactly the same thing? That's completely problematic, right? And a compounding problem was that people used to save their own data in their own little format, right? So different labs would have different ways of naming their files. And so if you were to say to some lab, hey, I love your results. I'm wondering if it's the same result as the thing that we're finding. Can I get your data? You'd get this pile of data that you would not know how to work with, right? Because it's named completely unfamiliarly to you. You know, they might be using different pre-processing steps. It's all a jumble. Yeah. So you can imagine that this is a really big problem for reproducibility, and it definitely was. So as a field, we're shifting towards this. And I want to highlight two things that have really improved the state of the field in this sense. So the first is the BIDS, the brain imaging data structure, which forces everyone to save their files in exactly the same way. And what we're also going to talk about in the practical Fmri Prep. So that is reproducible pre-processing. Yeah, so a set of people that are implementing like fixed types of pre-processing. And all of this is done completely openly. And so it's a really beautiful sort of open source, modern way of going about your research. So I'm going to quickly highlight these two things, especially Fmri Prep, the thing that I showed you as a second in that red box. That's going to be something in the practical. So what's this BIDS? Well, as your data come out of the scanner, it's a jumble of files like this. It's really hard to make sense of. So what the brain imaging data structure does is it says, even though your data might be coming out of the scanner in this sort of weird format, and different scanners actually have different formats, right? So it's really hard to compare across different types of scanners. What we're going to do is translate all of these files into one specific way of organizing it. Yeah? So this is no longer something that every lab sort of decides on for itself, but everybody does exactly the same thing. Yeah? And so then you just need to think about how do you go about saving your data? Well, let me point a few of these things out. So the brain imaging data structure is hierarchical. You see that for every subject, you have a separate folder. Yeah? So every subject is self-contained. If you add a subject, you just add a folder and you start populating that folder. You see that depending on the type of data that you put in, anatomical, functional, or diffusion weighted in this case, you have also different folders. And then for these different in these different folders, you end up with different types of files. Well, you have, so you have Nifty files, the red ones. So that's .nii.gz. These are gzipped Nifty files, and that's the standard file type for fMRI types of files. All sorts of metadata are implemented in JSON files. So you see here, these JSON files, these are text files, and what do they look like? Well, they look like this. So they're text files, and they just contain all sorts of metadata. So for example, what was your TR? What was your echo time? What was your flip angle? All sorts of things that may be necessary later on, information about how the data were acquired. Those are all put in these JSON files. And so the idea is that with just a Nifty file and one of these JSON files, you know everything you need to know about these data. You can then do anything later on. Now, anything else, things like behavioral outputs or a list of the characteristics of the participants in this case, those are put into tab separated files. So like nice tables. So this is a full description of how you should be saving your data, and people maintain it. And how do you know that everybody's using exactly the same format? Well, there's actually a validator on the internet. You can point it to your data set. Like, so you've done the work to ensure that it's in this BIDS format. You can put it through this BIDS validator at this URL. And this website can then tell you whether your data set conforms to the specification. It will give you exactly what are the errors and what are the warnings. So you can solve those, and everybody can then sort of iterate towards getting their data in exactly the right format. So this validator actually serves as a sort of gatekeeper. And once you fix it, then you can share your data with anyone, and they will know exactly what to expect. Right? Because that is the power of this sort of thing, right? For now, you're just thinking like, oh, why do I need to do all this extra work, right? That would be a shitty thing to have to do. The thing is, once everybody in the whole wide world stores their data in exactly the same way, you start to see where we're going, right? It means you don't care anymore whether someone at Princeton or someone in Melbourne or someone in Beijing acquired these data, right? You have, if you download the data, you have the data and you can work with them immediately. It doesn't matter anymore who or where this was acquired. So once you have this BIDS format, you can analyze everything. So it means that you can automatically analyze a whole bunch of different data sets. And this is what people have started to do. So they've started to create BIDS apps. So this is like an app that contains an operating system and a whole bunch of software inside it. You can point it to a BIDS data set, run a single command, and it will analyze that data set. Yeah. So because we can now expect everything to look exactly the same, you can automate much, much more. Yeah, and this creates a whole set of very, very powerful tools. Here's just some examples. You can have, we're going to look at quality control. We're going to look at FreeSurfer, which is this surface segmentation thing. We're going to look at Fmri Prep, which is this pre-processing pipelining thing. But here you see that there's a lot more. There's like tractography, there's cortical thickness analysis, and all of these just work, right? Because they can assume a specific outline of data. You see how powerful this is. It doesn't matter anymore that someone programmed it in their specific way. No, you just point it to a BIDS data set and it's done. Right? So if you want more information about this or to follow some tutorials, especially if you're going to, like, for example, run an internship somewhere, this is really what you need to know. Like, many labs don't do this sort of thing yet, but if you as a PhD student or an internship student can just implement this, you're going to empower that lab incredibly. I want to highlight one other thing. If you save everything in this BIDS format, this allows you to go through quality control problems much, much faster. So I've been telling you that fMRI data is really crappy. And this was up until now only the ways in which it is predictably crappy. However, fMRI data is also unpredictably crappy. It means that in principle, you have to go through all of your scans, visually inspect them, and quantify their quality in many different ways. And in the practical for this week, you'll go through a bunch of different quality assurance quantifications to understand how things can be crappy. Because there's a lot of problems that fMRI data can suffer from, and they're really, really hard to catch sometimes. So one of these BIDS apps actually automates the process of going through all of these, all of the files for quality control, and you're going to go through an output of this MRI QC. So just to highlight a bunch of these, if someone moves during the acquisition of the anatomical, you get problems like this, right? And so you really have to go through all of your anatomicals and visually inspect them. There's problems with lines in the images. So if something is in the scanner that acts as an antenna, it will pick up frequencies only at one specific frequency, and that can then translate into these really weird sort of geometric artifacts, or something like this, where there's like 1 or 2 points in K-space that get like blipped, and you get these strange periodic signals running through your scans also. And one other artifact of the idea that you measure these things as signals in time, and then you perform a Fourier transform on them, or inverse Fourier transform on them, is that you can get these ghosts. So you see like a second version of a brain outside of the brain, actually. So all of these things you want to be able to catch, right? Because it means that your data is not as good as you were hoping it would be. And this MRI QC is one package that allows you to go through a data set and then immediately inspect what it looks like. So you'll be going through that in the practical. You'll also be going through an Fmri Prep example. Let me just switch to just talking to you. And so you're going to get a feel for how powerful this is. What I've done is I've prepared a few sort of website outputs of MRI QC and Fmri Prep, this pre-processing pipeline. And that will allow you to not just get a feel for what these data sets look like, but you really start to see how different subjects behave differently in terms of the Fmri quality, the different things that can go wrong. And this is really something that you get a lot of sort of practical ideas about how does the data look. You really get an intuitive feel for what is a data set, so to say. Yeah, it seems like I'm at the end of my presentation already. I thought this would last a lot longer. What I want to say is, again, we're not like grading the practicals, but that we've tried to set up a brief run through of the outputs of this MRI QC and Fmri Prep. So with this background of the different pre-processing steps that exist, you should be able to consult the MRI QC website and the outputs that we've put online, and then understand exactly what's going wrong in what subjects, in what way. That's the idea. So if you end up doing an internship in Fmri, this is really the sort of thing that I want to say, this is the modern way of going about it. And if you ever end up in a lab that isn't doing this sort of thing, you can teach them. Because this is the way in which we as a field start making our science more reproducible. And I think that is, like we all know, right? Things with p-hacking and people, what is that called? Harking, right? Hypothesizing after results are known. That sort of thing you really want to guard against. And if everything is standardized in this way, as a field, we can start guarding against that sort of thing. Yeah. So thanks for your attention. If you have any questions, you can ask them now. You can, yeah, you can keep looking for data, like data dredging also. There's a bunch of different things that that are wrong right now that even though having open data and having open methodologies doesn't necessarily directly solve that specific issue. It is always a prerequisite for solving these problems in the future. So we're trying to give you some exposure to the way that things are done nowadays and the way that they should be done in the future. If you have any questions about the practicals, you can ask me over email on Canvas or wait until Friday and we can talk about it. I have not much more to say.
A question to the practical, will the video be uploaded soon?
About the one from the practical.
Yeah. Last Friday.
Yeah. What I've done up until now is I've uploaded the videos of the practicals to Canvas because I'm too ashamed of all the things that I'm saying in those. And these are uploaded to YouTube.
Okay. Yeah, I didn't know that the practicals are just uploaded to Canvas, but I'll have a look.
Yeah, I'll link to that I'll update the syllabus. Yeah, and like I said in the assignment that I sent around about the practical, right? Things are different than we planned, right, with this course because we're trying to make it more, sort of interactive. This means that things have changed also in terms of grading, and I understand that that is sometimes a bit confusing in terms of, okay, what is Thomas expecting us to do exactly? So I am creating something of an overview of that so that becomes a bit more clear.
Nice, thank you.
Okay, then.
Thank you.
Yeah, no problem. Thank you.
Thanks. Bye.
Bye-bye.
Bye.